And I'd like to thank you too, Robert, for the invitation to speak this evening. I'm going to talk briefly about what we found as far as the effects of chlorpyrifos and anxiety behavior, and what the zebrafish model can tell us about pesticide exposure during development. Chlorpyrifos is a kind of organophosphate pesticide that is classified as a neurotoxin. It's found in a large variety of foods such as corn, apples, peppers, grapes, plums, broccoli. 60 million pounds of organophosphates are applied to 60 million acres every year in the United States, and they've been in use since the 1940s. There are several routes of exposure to pesticides, mainly through ingestion of foods treated with pesticides. But they can also be ingested through the skin and through inhalation, through spraying of crops with the pesticide. Organophosphates have been found in dust and air samples in both homes and in daycare centers, illustrating that they are highly ubiquitous in the environment. Just to illustrate this further, this was a study done in children in which they were cycled through periods of conventional foods and then given organic diets to determine what the metabolite levels of organophosphates were during these periods. What was found is that children fed organic diets had fourfold lower or nearly undetectable urinary metabolite levels compared to periods of conventional food consumption, showing that food exposure is the main source of organophosphate metabolism. Most of you know the benefits of using zebrafish to study toxicology. Exposures can span multiple developmental time points over a very short period of time. And as far as our lab, we were able to analyze behavior by 7 days post-fertilization. So we work with larvae in particular. So the question that we wanted to ask was, does chlorpyrifos affect anxiety behavior in zebrafish larvae? And there were two reasons why we wanted to ask this question. One was that chlorpyrifos has been known to affect anxiety behaviors in the rodent model. And secondly, we have an assay that we developed in our lab that is useful for the detection of anxiety behaviors in zebrafish larvae. So I'm going to walk you through the novel high-throughput behavioral assay that we use in our lab. We use time-lapse photography to take images. For this assay is every 30 seconds. We have since modified that to every 6 seconds. We have a digital camera that is placed on the top of a cabinet. It's a wooden cabinet, and it's facing downwards to the bottom of the cabinet where we have a laptop placed with the screen facing up. On top of the screen can hold up to 4 multi-well plates. Beneath the multi-well plates, we can play a PowerPoint presentation to the larvae that we use as an aversive stimulus to provoke certain kinds of behaviors. After we collect our series of images, we can import the stacks into ImageJ, where we wrote a specific macro that is automated. It splits the colors into separate channels. We can subtract the background, apply a threshold to identify the individual larvae, and then this can give us XY coordinates of where the larvae are in the dish at any given time. For this assay, this is the PowerPoint that we used. At the top of the well, and this is a horizontal plane, not vertical. So at the top of the well, we have the moving, which we call the bouncing ball. And at the bottom, we have a stationary ball. The moving ball is the aversive stimulus that the larvae respond to. The bottom is just used as a stationary control for color in the well. So larvae that have been untreated, wild-type larvae, respond to the moving ball by moving down in the dish. So like I said, that's a horizontal movement. So they're moving away from the moving ball to the bottom of the dish. And they also move towards the edge. So they prefer to be on the edge of the well compared to the center when they are shown this visual stimulus. Like many other studies indicate, ligmotaxis, or the preference for the edge, can be an indication of anxiety behavior. And we wanted to follow through with this and look at the drug diazepam, which is Valium, known to have anxiolytic properties. We wanted to see how the larvae would respond to this treatment. Keep in mind that this was only given for 2 hours right before our behavioral analysis. So this was a very short acting period for them. So when we looked at the percentage of larvae down in the dish, the ones treated with diazepam looked exactly like our control treated with DMSO. So basically they moved away from the ball down in the dish just like the controls did. However, if we look at the percentage of larvae on the edge of the well, what we saw was that there was a significant decrease in the preference to be on the edge, both without the visual stimulus and with the visual stimulus. So the first half of the assay, we just have a blank white background for them, used as a control, and then the second half of the assay is where we show the moving ball. And for both of these, the preference to be on the edge was significantly reduced. So to follow up with this, we also wanted to administer a drug that has anxiogenic properties to the larvae. So we administered caffeine to them to see if this would also affect thigmotaxis behavior. And what we found, this was a separate assay, different from the previous one I showed you. But again, the larvae treated with diazepam had a significantly lower preference to be on the edge, whereas the ones treated with caffeine had a significantly higher preference to be on the edge, indicating that our assay was useful for the detection of anxiety-related behaviors in zebrafish larvae. So this brings me back to my original question was, does chlorpyrifos affect anxiety behavior in zebrafish larvae? Here's our exposure paradigm. We had 4 treatment groups of the chlorpyrifos and then the DMSO control. We started treatment at 2 hours post-fertilization all the way until 7 days post-fertilization in which the behavioral assay was performed. During this time, we also performed visual inspection for deformities of the larvae. We did find that the group treated with the highest concentration of chlorpyrifos, which was 1 micromolar, were malformed. So they had shortened tails that curved upwards, and they exhibited twitching behaviors, and they did not swim normally. Therefore, this group was excluded from analysis for behavior. However, we still had 3 other treatment groups of chlorpyrifos that looked identical to the DMSO controls. Therefore, they were still used for behavior. We performed 2 separate behavioral assays for this experiment. Like I told you before, the 5-fish assay, this one we had 5 larvae in each well of the multi-well plates. First half an hour we gave the blank background to serve as a control. The second half an hour we gave the visual stimulus with the bouncing ball. And then we quantified avoidance behavior, which is up or down in the dish, or thigmotaxis behavior. We also performed the 1-fish assay. This was an assay in which no visual stimulus was given. It was just used as a control to determine baseline speed levels of all the treatment groups. The results of the 5-fish assay. Here on the left is the results with just the blank background, and then the right is the second half of the assay with the moving ball. And what we found was that in larvae treated with the chlorpyrifos, they preferred to be on the edge significantly less than the lowest treatment group of chlorpyrifos or the DMSO control. So the 2 higher treatment groups of chlorpyrifos significantly changed their preference to be on the edge. When the visual stimulus was given, the highest treatment group, the 0.1 micromolar, showed a preference to be on the edge significantly less than the other groups. We did not find any changes in the percentage of the larvae to be down in the dish compared to the controls. As far as the 1-fish assay, without any visual stimuli, again we saw that the larvae treated with the 2 higher treatment groups of chlorpyrifos preferred to be on the edge significantly less than the other groups. Percentage of the larvae down in the dish did not change, but we also found changes in swim speed for the 2 higher treatment groups of chlorpyrifos. So just to recap what I told you, Larvae treated with the 1 micromolar chlorpyrifos showed deformations in their tails. The two other highest treatment groups, 0.1 and 0.01 micromolar, showed that larvae were on the edge of the well less of the time, and they swam slower compared to the DMSO control or the 0.001 micromolar groups. However, there were no changes in the percentage of larvae that were down in the dish at any time. So like I showed you in our assay, we believe that thigmotaxis is a measure of anxiety. So we believe that if larvae are treated with this pesticide during development, it definitely affects their anxiety levels. Motor speed is also affected. This could signify that some of the similar neural pathways are affected by the pesticide that affect anxiety and motor speed. However, avoidance behavior, because it was not affected, could be because there's a different mechanism involved with this behavior that is not affected by chlorpyrifos treatment. In the future, we would like to rethink health and safety guidelines for food in pregnant women and children, and should we be thinking about a larger push for organic foods? While the chlorpyrifos exposure did decrease the anxiety levels, it still signifies that there are some neural changes going on in the fish, which is a huge problem. And we don't know what happens in adulthood in our assay, but other studies show that there are differences between larval behavior and adult behavior. One of the next things I want to look at is we have a transgenic line that expresses GFP in neurons, and we want to look at the habenula, which is an area correlated with anxiety-related behaviors. So I'd like to treat some of these larvae with the chlorpyrifos and determine if the volume of the habenula changes or the number of neuron changes in these fish. Right now, I am currently looking at the different times of susceptibility during development, so I want to try to narrow down which specific days the larvae are most affected by chlorpyrifos treatment. And we also have improved our behavioral assay, in which now we have lanes that the larvae can swim in, giving them a larger swimming area, and we can quantify a larger variety of behaviors in the zebrafish. So with that, I would like to acknowledge my PI, Robert Creighton at Brown University, former research assistant Sean Pelkowski, our collaborator Ruth Cowell, undergraduates in our lab, and the NIA